Okay, today we're going to be talking about the cell cycle. This is a replication process known as mitosis. I like to remember mitosis by thinking of the T as we're going to make two cells when everything's all done. So first we're going to discuss the reason we do cell replication or mitosis, and then we're going to review the process. So why do cells replicate? First one is growth. So an organism will use replication to add new cells so they can grow in size. So an example, you start off as one cell after conception. You get half of your genetic information from mom and half from dad. So 23 chromosomes and 23 come together to form one unique cell that has never existed before, and that is you. And then by the time you're born, you have millions of cells. And then by the time you are an adult, you have trillions of cells. So you go from one cell, and as you get bigger, you add cells. Second is repair. So organisms use cell replication to repair damaged or old cells. An example is growing new skin for a cut, or so if you got like a bruise or a paper cut, your body's going to fix that by making new cells to replace the damaged one. The other one is you're going to replace old cells. So, for example, your blood is replaced every week or two. Other cells take longer. Other cells might only last a day or two, depending on what type of body cell it is in your body. Reproduction. Now, if you remember back in the beginning of the school year, we talked about the characteristics of life. So there's two types of reproduction. There's sexual reproduction, which requires a mommy and a daddy. And then there's asexual reproduction. So this is basically where it is used in bacteria and very simple organisms. You go from one cell, it is going to duplicate that cell and then split into two cells. So you go from 1 to 2. It is a very efficient type of reproduction. So for bacteria, some species can reproduce within 20 to 40 minutes. Now, whereas a human, it takes 9 months. So if it works for the bacteria, they're going to keep on doing it. So step 1 in mitosis. This is called interphase. So this is when the cell is just doing its normal function. So it is going to spend over 90% of its life in interphase. The chromosomes are replicated and they doubles the amount, okay? And this is where it's just starting to get ready for things. Cells will be in this phase for most of the time. So just to kind of give you a refresher, so I have my cell. So I have the outside, the cell membrane. I have the nucleus where the DNA is kept. And then I have those cell organelles around here. Everything we're going to be talking about today is going to be happening here to start off with. So here's an example of a cell under magnification. So I still have my cell membrane, I have my nucleus, and I have the organelles around here. Step 2, mitosis. So this is where I'm going to start going from 1 to 2. The cell is no longer functioning normally. The cell is starting to divide. So the cells at the end will have the same number of chromosomes. So depending on the species, they will have different chromosome numbers. For human cells, we have 46 chromosomes. So when we start off with one cell, we have 46. It's going to replicate or double that. But when it all is said and done, when I get two cells, I'm going to have 46 and 46. Now, if you had an organism with 12 chromosomes, it would start off with 12, and then I would get two cells with 12 each. Prophase. The DNA wraps into chromosomes. So the DNA is packaged here in the nucleus. It is going to wrap into, in humans, it's going to look like X's. So I'm going to get these X shapes. So it's going to kind of start unwinding. So here I have a plant cell, and you can see the outside of the cell wall. This lighter pink is going to be the cytoplasm and with the cell vacuole and stuff. But here you can start seeing all the genetic information unpacking. Okay, it's getting ready to do its business. Metaphase. The chromosomes line up in the middle. Okay, so we're going to start splitting everybody up. The way I remember is M for middle. So they all line up in the middle and they're getting ready to get pulled to each end. So in this picture, you can see the cell. You have the outside cell membrane. And then you have all the dark, that's the chromosomes lining up to the middle. And these lines are the centrioles. We're not going to really get into that, but they're kind of like ropes, and they're going to pull the genetic information to each side. So, anaphase, or axed apart. The chromosomes move away from the center to the sides. So remember, they were all lining up here in the middle with metaphase. Now they're getting pulled to each side. So you can either remember A for away or A for axed apart. Whatever's going to help you remember that. Telophase. The DNA unwrapped into a pile of DNA. So basically, this is what sometimes looks like a peanut. So I'm going to start getting what's going to look like the two cells. The DNA is going to unwrap and uncondense, and the nucleuses start to reappear. So I'm going to start seeing what looks like a peanut with two nucleuses inside. And then what's going to happen is the cells are going to pinch in the next phase, which we're not really going to talk about, but they're going to pinch, and then I'm going to get two cells. Telophase starts to see two cells. So here in an animal cell, I have the outside of the cell membrane, and I have the DNA in the middle with the nucleus, and I can kind of see that middle part that's going to happen. In a plant cell, it's going to be more squarish. I have the cell wall. Right below that's going to be the cell membrane, and I'm starting to get the two cells, and I got my two nucleuses again. And that's it.